Me, María del Mar Amezquita, and my partner Santiago Alvarez, medicine third semester students, are going to talk about how EBV-miR-BART5-3p promotes the proliferation of Burkitt lymphoma cells via glycolytic pathway.
What does the microRNA do? The microRNA helps Epstein virus to evade the immune response and the survival cell. How? First, inhibits apoptosis, then promotes the viral latency, and finally it contributes to oncogenesis. The Burkitt lymphoma is a highly aggressive from B-cell lymphoma that primarily arises within or after germinal center. Burkitt lymphoma accounts to the 30 or 50% of childhood tumors in Africa. It's reported by the World Health Organization. Burkitt lymphoma has been categorized in endemic, sporadic, and immunodeficient associated. The glycolytic pathway is a fundamental metabolic process in which the glucose is broken down into pyruvate to produce energy.
The microRNA attacks TP53 gene inside the cell. The decrease of this p53 gene increases the hexokinase 2 enzyme, which is essential to processing sugar, and removes the brake, that is the p53 gene, on the GLUT channels, allowing a lot of glucose to enter the cell. So lactate is produced massively as a product of glycolysis, generating uncontrolled growth and energy. Those are perfect conditions for the growth of a tumor.
The principal objective to this article is to investigate the role of Epstein virus in the pathogenesis of Burkitt lymphoma, specifically how this viral microRNA regulates glucose metabolism in the glycolytic pathway through the inhibition of TP53, thereby promoting cellular proliferation and survival.
Western blotting is a technique that is used to identify and qualify the exact amount of some protein inside the cell. The scientists will use a chemical substance to break the cells and make a lot of their proteins out. Then, they will separate them by size with a special gel and electricity. Then they will use some markers which will stick to the specific protein they are looking for. And finally, it will be seen as a bright amount of mark. It's used to precisely measure the activity of certain genes in cells. First, RNA, the molecules containing genetic instructions, is extracted and converted into a more stable form of DNA for analysis. Then, using the qPCR technique, millions of copies are made of the specific fragments to be studied, such as viral microRNA or the protective gene TP53. During this copying process, a light-emitting dye is used, allowing a machine to measure the intensity of the light to calculate the original quantity of that gene, always comparing it to a standard reference, would be GAPDH, to ensure accurate results.
Su principal objetivo fue, primero, aumentar o disminuir el Epstein virus. Para eso se utilizó los plásmidos que estimularon e inhibieron para así comprobar sus niveles altos o bajos en las células. Lo segundo fue evaluar el efecto biológico del microARN. Tras la transfección se pudo analizar la proliferación y migración, su actividad glicolítica y cambios en la TP53, y así se comprobó que el Epstein virus promueve el crecimiento y la supervivencia celular. Y por último, confirmar la relación que tiene el microRNA-BART5-3p con la TP53. Luego de que se modificara el microARN, se usó la RT-PCR y el Western blot para ver que al aumentar el Epstein virus se disminuye la TP3 y al inhibir el Epstein virus se aumenta la TP53. Esto nos dice que el microARN es el que regula la TP53. Este se realizó para evaluar cómo la EBV afecta la proliferación de las células del linfoma de Burkitt. Y se logra evidenciar que, primero, que la EBV aumenta la proliferación de las células del linfoma. Los resultados mostraron que su sobreexpresión incrementa la proliferación celular, mientras que si inhibía se reducía. Con esto se apoyó la idea de que la EBV tiene efectos oncogénicos en esas células. Y segundo, la relación de la proliferación asociada a la TP53 y glucólisis. Gracias a esta prueba CCK8 se vio que al inhibir la TP53 y examinar la glucólisis se veía un aumento de la proliferación celular.
Here, the researchers tested what happens if we manage to switch off the virus stool. By using an inhibitor against the viral component, the patch of the protective protein P53 regains strength and thickness. This is crucial because it proves that if we slow down the virus, the cell regains its natural ability to defend itself and stop tumor growth. Then we have this other experiment that demonstrated the cell's ability to move. Each blue spot you see is a lymphoma cell that managed to migrate through a filter. You will notice that in the group where the protective protein was blocked, the number of blue spots is much higher than the other one. This visual test shows how the virus activates the cell's energy machinery. You'll see that the patches of the HK2 and GLUT1 proteins, which function as the engine and gateway for sugar on the cell, are much darker and thicker on the cells that have the virus microRNA. This demonstrates that the virus forces the cell to produce these proteins to feed on sugar and grow aggressively. The line below, GAPDH, is just a control to confirm that the comparison was fair and that there were no errors on the sample.
Este gráfico acá básicamente se evidencia como la proliferación celular. Acá se utiliza el EDU, que el EDU se incorpora a las células que están en la fase S del ciclo celular y se detectan con fluorescencia. En síntesis, la prueba muestra que en donde hay más fluorescencia, que podría ser este recuadro o estos, es donde hay más proliferación por la sobreexpresión del gen del EBV-miR-BART5-3p. Acá en este gráfico vemos la evidencia de otra prueba, que el objetivo principal era comprobar si la EBV tenía efectos directos con la TP53, si la disminuía o si la alteraba. Acá se evidencia como, por ejemplo, cuando la BART5-3p está estimulada, los niveles de P53 disminuyen en comparación de cuando están en condiciones normales. En este gráfico vemos otra prueba que el objetivo principal fue analizar la expresión de la proteína GLUT1 que está relacionada con el ciclo de la glucólisis. El objetivo era ver si la inhibición o la estimulación de la BART5-3p o de la TP53 la estimulaban o la reducían. En este caso, cuando se inhibía la BART5-3p se puede evidenciar que la GLUT1 bajaba su concentración, mientras que cuando se inhibía la P53, esta subía su concentración.
These are some opinions of the authors of this article, and my colleague and I think they are correct, because their arguments are consistent and prove points from the article itself. For example, it is irrefutable that the TP53 is considered one of the most and well-known cancer suppressor genes on the earth, and that the protein of study in this case plays a crucial role in glycolysis, as I explained earlier with the diagram.
For me, first, the molecular biology opens the door to the new and more effective possibilities in medicine, making the treatment for chronic or terminal disease more efficient. The second conclusion is in the future, thanks to molecular biology, we may be able to find a cure for diseases to currently have no prognosis, such as cancer and other diseases. The only problem that I see is these genetic therapies are very expensive and become unaffordable for most people who don't have financial resources.